actually works.
So you have a spectrum of 3 fascinating talks that at the first glance may look very unrelated. But there are relations. There is the science, the weave of science that keeps things together. So please.
Hi, I'm Willow from Florida, and first of all, thank you for the very amazing talks. They're mind-blowing. My question is for Francis Arnold. I wanted to, at some point in your talk, you mentioned that the poor-performing proteins, they were originally just tossed in the garbage, and then you mentioned that now that they're used for machine learning algorithms. Can you expand on that?
Sure. What are we trying to do? We're trying to understand the mapping from sequence to function. It's a complex map that we cannot design. We don't understand it, but it exists. So machine learning is very good at finding those complex patterns, as long as you have the data. And in directed evolution, we have a lot of data, and also the databases are getting filled with protein sequences, and even sometimes some annotation as to what those sequences do. So the idea is now, instead of just throwing away the ones that don't work, we take all of the data and feed that into machine learning algorithms, which can tell us how to design the next generation. So if we're going to make mutations, where should we make them? What combinations of mutations should we make in order to test a minimum number of sequences and jump higher in every generation?
Thank you so much.
Okay.
Hello, my name is Xena Lumet, and I'm from Brynässkolan in Fagersta. And my question is to Professor Arnold. Is there any risk concerning the mutations that are induced in the enzymes? Could it be threatening in a way to create enzymes?
Could be threatening?
Yeah.
Well, enzymes are not self-reproducing, so that's one nice thing. If you've got enzymes in your laundry detergent, they're not going to get out and attack you unless they're badly designed. So it really depends on how you use the chemistry, right? You could use the chemistry just by itself. The enzyme is a catalyst that uses earth-abundant metals, or you could put it into a genetically modified organism, which becomes like the poodle and is less fit. We don't make anything that's going to get out. We don't put any genetically modified organisms out into the environment. We just use the enzymes as catalysts.
Okay, thank you.
Thanks, Anna.
Hello, my name is Kylie Long, and I'm from Florida Atlantic University High School. And my question is for Professor Sandra Diaz. So I wanted to hear your opinion on the genetic modification of plants and what effect this would have on your the global spectrum that you were describing.
Okay, in theory, you can have genetically modified plants in any part of the spectrum. The genetically modified plants we have today are all concentrated in our favorite bit of the spectrum, because that's the kind of plants we like, genetically modified or not. But in theory, we could come up with genetically modified in all bits of the spectrum, including the bits which are in deep trouble now.
Okay, thank you.
Thank you.
So my name is Marwan. I'm from Talya Gymnasiet in Södertälje.
Please speak into the microphone so we can all hear.
My name is Marwan. I'm from Talya Gymnasiet in Södertälje. My question is for Professor Omar. If the water that is produced from moths, is it pure? If it is pure water, how is it possible that it is useful for humans, since humans can't really drink pure water in a large quantity?
Good question. So the water that you produce is ultra pure. It doesn't have minerals in it, but mineralizing it is quite easy. That's what municipalities do all the time. You can take a packet. It would be built into these devices that it would be mineralized and so therefore drinkable. But otherwise, you could use it also for agriculture, for many other applications. So the point, I think, is that the moth produces water that can be used for anything, including drinking if you mineralize it. Yeah.
Okay.
Thank you.
Hi, my name is Johanna and I go to Arnold Gymnasiet. And my question is for Professor Yaghi. What is the biggest obstacle that you face when creating the moths and how do you solve the problem?
The biggest obstacle, I think, was that nobody believed that it could be done. I have to be honest because I'm talking to the emerging scholars. And even after we showed that it can be done, there were a lot of doubters out there that introduced new doubts and new challenges. And every step of the way, there were these challenges that people put on moths. That first it was they're not going to be stable, they're not going to be porous, they're not blah, 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 all the way. And we persisted. But during this process, something very important happened. And that is that every time there was a bunch of doubters, there was one or two people who believed in what I was doing. And they made all the difference. So I think we have to persist in what we believe could be done. And it taught me that nature is so rich. So anytime somebody says it can't be done, it's very easy for you to prove that it can be done because there's so much out there that's waiting to be uncovered. You just have to give it a chance.
Thank you.
Hello, my question goes to Dr. Arnold, and thank you for the inspirational talk. And it's 2.5 parts to it, but I'll be very quick and fast. So the first part is with regards to your PNAS paper that was published 2 days ago about machine learning and changing 4 residues in a protein at the same time. Do you take sequence alignment into account? In other words, there are certain residues that are extremely well conserved. So when you try to make this enzyme into a new type of enzyme, do you take those residues into account?
Well, what's really interesting is that if you want novelty, very often that happens when you make mutations at residues that nature has conserved. So everything is possible when you explore the non-natural world.
Okay. And the second part was, have you explored using amino acid number 21 or changing base pairs? Because he might be a colleague of yours, but 10 years ago, Dr. Peter Schultz from the Scripps Institute at the 50 at ENC, he talked about introducing the 21st amino acid and so on, and he has a strain of bacteria that uses that. Yeah. So basically, he's trying to expand the genome and with all the tRNAs and things. Have you explored that pathway?
Yeah, that's a great question. So why limit yourself to the 20 natural amino acids? There's no good reason to do that other than it's technically difficult to do directed evolution by incorporating the non-naturals. That's going to be solved in 5 or 10 years. Somebody's going to figure out how to make it much more efficient. Then evolution can include whatever you want to use. The problem, of course, is that the possibilities grow exponentially. So you have to have a good reason to think that adding number 21 gives you a higher probability of finding the novel function that you're interested in. And that's a good chemistry problem.
Thank you very much.
Hi, my name is Ines and I'm from Soltorpsgymnasiet. I have a question for Professor Sandra Diaz. How long did it take to do the research to find all the data for the global spectrum of plants?
How much it took us?
Yeah.
Okay, once the database was, okay. I would say a total of 10 years, because the paper I showed to you, that took us about 5 years. But for that paper to be possible, we relied on a global communal database, which is out there, open to everyone on Earth, which it's a gathering of all the data of functional traits of plants contributed by all scientists who studied it, and organizing that network for us and for the rest took us another 5 years. So altogether it was a lot of work. The analytical bit, that was very easy. The big thing was really getting the database.
Thank you.
Hi, good morning, everyone. I am Onoya from Stockholm University. I have a question for Professor Arnold. Firstly, it's an amazing biochemical feat that you have done, and I mean, even by today's standards, it's not an easy task. But I was curious to know, how do you maintain the directed evolution, because I mean, for practical purpose especially, because we know nucleic acids have a tendency of undergoing spontaneous mutations. You know, there can be. I mean, I'm just a little curious to know about the technical part, if you could.
Sure. The spontaneous mutation rate is way too low to be useful if you want to direct evolution in a timescale that matters for your PhD.
My question, I'm sorry to interrupt you, but my question is, do you face the problem of spontaneous, I mean, once you have
is that a problem?
Exactly.
No.
I mean,
unless
for practical purposes.
So, yes and no. During the evolution, the spontaneous mutation rate is very, very low compared to the mutations that we introduce. But what happens is if the function that you're creating is antithetical to what the bacterium wants, it tries to kick out the DNA. But that's an old problem in synthetic biology and recombinant DNA, so you usually use an antibiotic selection marker to force it to keep the functional DNA.
Okay,
yes.
So you
use tricks.
Okay.
Good question.
Thank you so much. Thank you.
Hi, my name is Yi Cheng Chung from Nesbru videregående skole in Norway. And my question is to Professor Omar Yaghi. And what are the costs of producing moss? Because the people who need it are relatively poor, and so is it affordable for them to get that?
So MOFs have been, as I mentioned, scaled up to multi-ton quantities by chemistry giant BASF, is a German chemical company. And the cost is not publicly known, but it sort of scales with the cost of the metal. So if you have aluminum, which is dirt cheap, you can imagine that the MOF could be made cheaply. It also depends on how much you're going to use. And so if you're going to use 100,000 tons, I can tell you that MOF is going to be dirt cheap. The nice thing about the MOF, as I mentioned, is that it can stay in the device for the lifetime of the electronics, which is around 5 to 6 years. So you don't need to necessarily take it out every month or have a cartridge changed. And in fact, when the MOF doesn't work anymore, because it lost its porosity or whatever, after 5 to 6 years, you can go in and add a strong acid and separate the metal from the linker and then reassemble it. You could reassemble it into that same structure, or with some fiddling, you can assemble it into a new structure. So I think the cost is not an issue here. Yeah.
Okay. Thank you.
Yeah, you're welcome.
Hi, my name is Ebba, and I'm from NT Gymnasiet Uppsala. And my question is to Professor Arnold. And I was wondering if, and you mentioned in your speech that we cannot write genetic code ourselves. And I was wondering if you thought that we would be able to sometime in the future understand it, and if we could, if we should focus on that, or if it's pointless and there's easier ways to, you know, as you talked about.
No, that's a wonderful question. Should we keep trying to learn the rules of how DNA encodes function so that someday we can compose and not have to go through this expensive empirical process of evolution? And the answer is clearly yes. It's a human desire to want to do that. But we shouldn't forget that also we can use machine learning techniques, and so it'll be a race between those who would want to understand it sufficiently and those who can just do it statistically from machine learning. And maybe it's a combination of all of those things that will be the best.
All right, thank you.
Hello, My name is August, and I'm from Victor Rydberg Odenplans Gymnasium here in Stockholm, Sweden. And I have a pretty open question for Professor Yaghi. This water harvesting, can you talk briefly about what opportunities this presents in our effort to combat desertification?
So I think the main contribution of such a system would be to deliver water to regions that don't have water. And so, especially in the desert, okay? But also in regions that are humid, but you don't have clean water, you could also, these systems work extremely well under those conditions. But really where we need water is in the desert. And because of climate change, we heard yesterday that those regions are getting lesser and lesser water. So I think, I hope I'm answering your question. I think the key contribution here is that we have a way to take water from the atmosphere, concentrate it, and deliver it where we need it in the desert, or to people that need it.
Just a quick follow-up.
I think we have only time for one question each. We are running out of time. Please.
Hello, my name is Fenja, and I'm from Knuthansskolan in Ronneby. And my question is for Professor Arnold. And it's, what are your opinions about changing enzymes or DNA in humans?
Should we change enzymes in humans?
Yes.
So we're not going to be doing any directed evolution in humans. But if you can cure a disease with somatic editing, and you know what mutation to make, I think that's going to become a standard treatment for disease, if you know what you're doing.
Thank you.
Good morning, my name is Laura and I'm from IHGR, Gothenburg. My question is for Professor Arnold. Would it be possible to develop an enzyme protein sequence that is not found in nature at all?
Well, I have lots of them. So the sequences, nature's only explored a tiny fraction of the possible sequences. It's very easy to make a sequence not found in nature. But the real interesting thing is to make a function that's not found in nature. And clearly that's possible as well.
Thank you.
Hello. My name is Moa Linear. I'd like to thank you for being here. My question is directed to you, Professor Arnold. When you modify the original nature-made enzyme to do something new, does the active site change? If so, how?
That's a great question. That's called biochemistry. Biochemistry is hard. And it's also called reverse engineering. So an engineer has made a new device, a new enzyme, and you want to understand how did that new property come about? You go in and you tinker with it, you look at the structure, you do spectroscopy, you do biochemistry on it to try to understand that. That's actually a lot harder than making it in the first place. But we do that once in a while. And the stories are fascinating because the answers that come out are almost always a big surprise. It wouldn't be something that a simple monkey mind would predict, right? That's what we say, you know, what would you predict? When we make predictions, we find out the answers are all something completely different, which makes this whole idea of design even farther away.
Thank you very much.
Yeah.
Hello, my name is Sophia. I am from Västsvensk Gymnasium, Gothenburg. I have a general question. I asked many persons, but I never get my answer. It's about, okay, if we have already started working for globalization and greenhouse gases, we can reduce to release greenhouse gases, but can we stop it? And for example, if we can stop it also, it will be help us, or it will be not late for us to save our atmosphere or climate in the future.
Omar, I think you could start.
Well, I mean, I think that we have to use the resources that we have on our planet responsibly. So one of the examples that I gave where you can capture the CO2 and convert it to a fuel is one such example. So I think we have to start thinking in that way. The idea of taking them off and then taking it back to its original building blocks, and then reassembling is another way of using things, cycling things, and thinking about the lifetime of the material. Yeah.
You want to add something?
No, I think it's pretty much.
Yeah. Yeah.
Well, plants are a fabulous way of taking CO2 out of the atmosphere. And you can imagine a sustainable economy that's based on biology. You can imagine that. It might be a little bit hard to implement, but biology is already pretty good at taking CO2 out of the atmosphere. So if you can turn plants into other things, as humans have done for a very long time, you're doing that with your trees, right? Look at this room. And then you have a circular and cyclical and sustainable way of making what you need.
Yeah. Thank you so much.
Yeah.
Hi, my name is Nika Velasco. And I'm Nicole Rivera, and we study at Anna Whitlock's gymnasium right here in Stockholm. We have a question for you, Professor. Oh, sorry. We have a question for you, Professor Arnold.
Put it by your mouth. So yeah.
Should I just?
That's
Is this good?
Yeah.
Right. We have a question for you, Professor Arnold. The possibilities with this artificial selection seem endless, but what are the main risks and dilemmas? And could this technology be used for controversial purposes?
Yeah. I mean, this is always the question we face with new technology. And any new technology can be used for nefarious purposes, and we seem to have a particular worry about biotechnology. There's this yuck factor. We don't want to eat genetically modified foods, but there's no good basis for a lot of those worries. Because, I mean, look at how people have used, instead of having insulin from ground up pig pancreases, we use genetically modified organisms to make insulin, and no one complains about that. It's just that, you know, we have to get used to these technologies and make sure that the benefits outweigh the real costs.
May I add something to that? I think the real worry so far with GMOs is the technological package that comes with them. For example, most of the really big environmental problems with GMOs, typically, for example, soybean, don't come from the soybean itself. It comes with a pesticide package that goes with it. And that's really terrible, and that really impacts nature, and I think we should all worry about it. But it's not because of soybean bee GMO itself. It's the technological, basically the business model that goes with it.
Thank you.
Hello, my name is Marcus Boman, and I'm from Ova Gymnasium. And my question is for you, Professor Yaghi, and that is, would one of the more effective water extractors, like the one that extracts over 22,000 liters, have any negative effects on the environment it's acting in, especially if the area already is hyper-arid?
Yeah, I mean, I think you have to look at whether you would change weather patterns if this is applied, let's say, for the entire Los Angeles area. But I think that there may be ways around that where you can have your harvesters out of town, so to speak, and then you pipe the water in, or you have, different scenarios that basically you have the water uptake being done out of populated areas. But still, we don't know what kind of effect that would have on the atmosphere. But keep in mind that the water that's being used will also, it's recyclable, so it still will evaporate. And but that's something that ought to be studied if this is ever going to be applied on a much larger scale. But it would take a lot more than the 22,000 liters per day to worry about such problems.
Thank you.
Yeah, you're welcome.
Hi, my name is Regina Albatova, and I'm from Anna Whitlock's Gymnasium in Stockholm. My question is for Professor Arnold. If directed evolution were to be implemented in nature, what could the negative consequences be of that?
That's similar to the previous question, but we've already been doing directed evolution, right? I mean, there's lots of, the human footprint on the planet is enormous. When you wash your hands, you're doing directed evolution. So we do that all the time. We're just doing it with proteins now.
Yeah.
But those are the things that you have to worry about. And we should stop doing some of the directed evolution we're doing.
Thank you.
Yeah.
Hi, I'm Jenny Jonasson from Härnösands Gymnasium, and I have a question for Professor Arnold, and that is, after all the hard work you have done, how did it feel to win the Nobel Prize?
Well, it's still pretty new for me. I'm still, wrapping my head around it. But it feels pretty good. I can recommend it. And one of the benefits is that my children do their own dishes now. They don't leave dishes in the sink for me anymore.
Thank you.
Hi, my name is Amina and I'm from Härnösands Gymnasium. And I have a question for Professor Arnold and Professor Yaghi. And I was wondering if you have any specific goals you want to reach within the next 10 years. And if you do reach them, what will those accomplishments do for us?
You go first.
Well, I would say that in terms of the chemistry that we're developing, the goal is to implement this idea of sequence-dependent materials. Right now, materials work where you have one material, one function. But I think the idea of this heterogeneity within order is so powerful that potentially you could have a material that can direct molecules in specific circuitry, and along this circuitry, they could be transformed and change in ways that you can't do any other way. So basically, I'm talking about anisotropic reticular chemistry, where now you can direct things exactly where you want them within the pores, and along the way you will have stations, reactions, different mechanisms to allow you to carry out reactions, energy transformations, and many things that we cannot do right now. But biological systems can do very well. So this is a very nice way to say, perhaps instead of mimicking the biological system, this is a way for us to concept transfer from biology. So I would say I would like to see this heterogeneity within order idea developed further and bring it to fruition in terms of applications that are reminiscent of biology, but cannot be incorporated in artificial materials. So I would say sequence-dependent materials is one direction that I would like to go. Obviously, there's a lot of stuff about weaving, but I want to allow Professor Arnold to tell you her idea.
Hi, I'm Andrea Sartorius from Ova Gymnasium in Täby, some kilometers away from here. And I have a question for Sandra Diaz, and it is, can you give an example of a rare but viable combination of traits in a species, and why are these traits not favorable? Why are these in the outer space of your findings?
I apologize, I couldn't understand the question very well.
Give an example of where rare traits are combined, and why are those outliers there?
Okay, wonderful, thank you. I think examples, for example, combinations of really big seeds in small plants are rare. We also have comparatively few species in the world which are really tolerant to nutrient deficiency. I'm talking extreme nutrient deficiency. We are also very, we have very few plants with really big leaves. We are very scarce in plants which tolerate anoxia very well. Apparently, coping with drought is not as much of a problem as anoxia. They don't know how to do it, and very, very few lineages have to do it. And if you look at the fringes of the spectrum, they are all very specialized, and they are sort of cheaters. For example, orchids are in one extreme. They are incredibly stress tolerant. They grow very slowly, and they have tiny, tiny seeds. So how do they manage? And the answer is, when they are seeds, they require mycorrhiza, fungi, to germinate. Without their fungi, they are nothing. They don't survive. And in the other end of the spectrum, some plants which are really, really nutrient, they can cope with very low nutrient availability. They can do so, again, because they have special association with fungi, or because they are carnivorous. Being plants, they are carnivorous, so they manage to cheat the system somehow. Now, those specializations tend to have come into evolution only through some special families. And we believe that, first, the environments where they grow are not very common. And second, they are very unlikely. They have many problems in other places. I mean, if you take one of these really specialized plants and try to grow it in your garden, they will never grow. So that's why they are so rare. They are basically the losers of the massive selection process we are exerting on the planet. I fully agree with Francis that we are directing evolution all the time. We have been doing that since we became human. And these are the losers of our domination of the world.
Thank you very much.
Hi, my name is Victor Lindahl, and I'm attend Åre Gymnasium here from Stockholm. And I have a question for Professor Arnold. Is it possible to clinically create new enzymes, like that you just take the parts or the sequences that you want to, or do you have to do it in breeding, or would you gain any time, or instead that you do it clinically?
I'm sorry, I didn't quite catch the...
You use breeding
Yes.
for creating new enzymes to do a certain thing. Could you clinically just take an enzyme and put together a new one instead of doing the whole breeding process?
So very often, a good breeder always starts with a good parent stock. So if there's an enzyme out there that's close to what you want, that's a good place to start. You don't get a racehorse from a donkey, right? So you want to start with something close to a good racehorse. So that's your job as the molecular breeder, is to figure out, given the functions that you want to achieve, who is the parent. And might be an enzyme you already have in your refrigerator. So I look at those kinds of problems. If you want to combine enzymes, maybe you get two functions at once. Yeah.
Thank you.
Hi, my name is Christian. I'm from Gymnasiet in Södertälje. I just have a doubt for Professor Yaghi. Might sound a bit fantascientific, but you said that your materials that you're using have a way higher capacity than a normal tank. So might them be used to transport water or other gases on another planet, I mean on Mars, and recreate an atmosphere there? Because on Mars, they think that there was water there once. So might it be possible to recreate an atmosphere there?
That's a very good question, very insightful question. In fact, I know that some people are working on that problem, is that there are places in Mars where you have ice that is not where you want to eventually live, I guess, or create a station. So what they want to do is create a vapor pressure by melting the ice and then store the water, then move it to the location where they want to build the station. So yes, I think once you start thinking about the possibilities of what this could provide, and I want to emphasize the mobility, this new mobility that you have, and the applications are almost endless, and that's just water. But think about hydrogen storage and methane storage and gas storage in general. That's just those MOFs where we're just designing the backbone. We don't even have the heterogeneity that has such a tremendous potential yet. And we are talking about foundational applications that will change how we do things on this planet. So thank you very much for that nice question.
I have another, really small question. Talking about Mars, there the concentration of carbon dioxide is really high. So might it be possible transporting there a system that takes this carbon dioxide and it transforms it into fuel?
Sure.
So okay.
Sure. I think this is Planet Earth Symposium, so maybe we should have a Planet Mars Symposium. But yes.
Okay, thank you.
Yeah, thank you.
Hi, my name is Erik Hollander, and I'm from Ova Gymnasium here in Stockholm, and I have a question for you, Professor Arnold. There's a debate in the US on whether or not intelligent design...
Sorry, you'll have to speak.
There is a debate in the United States on whether or not intelligent design should be taught in US schools. Yeah. What would your go-to argument be for evolution and to debunk intelligent design?
Yeah.
Would you mind sharing?
Obviously, I'm a fan of evolution. There's real data supporting it, and there aren't any data supporting intelligent design, so I don't support the teaching of intelligent design.
Thank you.
Hello. Well, my name is Ludwig from Tullinge Gymnasium. I have a question for Professor Arnold and Professor Yaghi. Professor Yaghi talked about how these MOFs have some similarities with organic molecules and DNA. And I wonder if it would be possible and or meaningful to, so to say, combine your two areas, find a way to make them, so to say, replicate and apply evolution on the MOFs. Yeah.
Yep. One of his PhD students is coming to work for me. We're going to do that.
Let me just say that the hidden agenda behind heterogeneity within order is ultimately to be able to do what you just suggested. You can't do it with the backbone, but you can attach that stuff that's going to do all the dynamics, replication, all the things that you want. You can attach it covalently on there, and now it is free to carry out these functions within the containment that the backbone produces. And so that's a very nice question. Thank you.
Thank you.
Yep.
Hi, I'm Sophia from Victor Rydberg Odenplan at Stockholm, and I have an open question on whether you have any advice for people in the audience who want to become scientists like you.
I think each of you should address. Start with Omar.
I have a lot of advice, but one thing I would say is that think about any problem that our planet faces, whether it's environmental, climate, disease. Who's going to solve that problem? Okay, it's the scientist. Everybody comes back to the scientist. And we're only less than, I don't know, researchers in science are probably less than 1% of the global population. So it's almost our duty to be scientists, because those are the people that solve the real problems facing the world. If we leave it to the accountants and the others, not as effective. The scientists can do it.
Follow your own path. Don't follow the herd. If you trained like everybody else, you're going to think like everybody else. So do what you find interesting, and maybe you'll put it together in some new way.
And I would say follow your passion. There's a very large number of problems you could solve. Many of them are urgent. You cannot possibly address all of them. So think of which one arises your passion more strongly. Because there is where you're going to find innovative questions. If the problem is very useful but bores you to death, then you are not going to find anything useful or innovative.
Thank you all very much.
Hello, my name is Clara and I'm from Furuhögskolan in Kalix. And my question is for Professor Arnold. How would you say that the directed protein might solve the expected famines in the future?
How directed protein might
solve the expected famines in the future.
Expected?
I might be using the wrong word, I'm sorry, but like hunger that is expected because of the growing population.
A famine. Actually, I'm working on that. One of my biggest projects and efforts is to do sustainable agriculture and replace pesticides entirely with mating disruption. Insects find each other with molecular plumes. So if you spray their perfume in a field, the male gets confused and he flies around and he can't find her, and then he doesn't mate and they don't have caterpillars. And this is a completely non-toxic, organic way to control pests. The problem is it's a chemistry problem because these perfumes are really expensive to make. So if you can make them cheaply using biology, you could actually use this and we have field trials in Africa now to combat the fall armyworm infestation using these insect pheromones. So I think there really are real things that you can do with this to work on that huge issue.
Thank you so much.
Hi, my name is Adam from Nacka Gymnasium here in Sweden, and I have a question directed for Professor Yaghi. So yesterday, Professor Haig talked about the fact that we need to sort of suck out carbon dioxide from the atmosphere in order to reduce the effects of the greenhouse effect. So would you consider MOFs to be an effective way of combating the greenhouse effect due to the fact that it can trap carbon dioxide?
Yes, that's one of the solutions that I'm proposing. By binding those primary amines covalently onto that backbone, right, you can selectively pluck out CO2 out of a mixture of gases. And that's the kind of selectivity that you need to implement that vision. Mm-hmm. So I showed preliminary results on that, but there's a lot of work that we have done in this area. Perhaps in future there would be another nice symposium like this dedicated to CO2. Yeah.
Thank you.
Yep.
Hi, my name is Emily and I'm from Katedralskolan in Lund. And my question is for Professor Arnold and Professor Yaghi. And I guess in a way my question has already been answered, but I was wondering if you had any fields outside of your area of expertise that you would want to expand your work into. So like, for example, you talked about combining your two fields of work. If there are any others you haven't mentioned yet that you would want to work with, perhaps.
I'm sorry, I didn't quite.
Fields do you want to expand outside your expertise?
Computer science. I think machine learning and AI are going to be invaluable tools in helping us discover and discover new structures, new reactions, new mechanisms. So it's going to be a powerful, we need to modernize. Chemists need to modernize and use these as tools. Yeah.
I'm always working outside my expertise. So everything I do is outside my expertise. That's the great thing about science. You learn something new all the time.
Okay, thank you.
Okay, my name is Annabelle from Donner Gymnasium, and my question is for you, Professor Diaz.
In what ways do you think agriculture should, like in what direction should it move in order to restore what you call the fabric of life?
Okay, good question. I think we have to do two things. One thing is, it's like with a disease, right? If you just jump from the roof and have all broken bones and internal bleeding and everything, you need to do two things. One is deal with the symptoms immediately, but obviously just dealing with the symptoms is not going to take you very far. You know, you need aspirin, you need painkillers, but also you need the broken bones fixed. So with the unraveling of the fabric of life, we of course need to have some urgent fixes. So for example, we need to create more protected areas in key areas of the world where some really threatened species are. We need to set up more programs to deal with the threatened species, etc. So we need to do that, but that alone is not going to take us to there. What we need to do is tackle the root causes, and the root causes of what's going on with the fabric of life is our way of doing business, our way of running the economy, our lifestyle. We all have these social narratives into our heads that says, in order to be successful as a person of the country, you have to consume more and more all the time and dispose more and more, faster and more. And that mental program in countries and with people is the root cause of the problem. So we need to make sure we build considerations for caring for the fabric of life into sectors of society which are not the environment. So just having better environmental policy in the environmental minister will not take us there. We need that, but we also need to build biodiversity considerations in the economy. For example, if new roads are constructed, are they good or bad for biodiversity? And if they are bad, they have to be changed. And if they are neutral, can they be good for biodiversity rather than neutral? The same with the way in which we produce clothing, the same in the way we produce food and we dispose of the waste. The subsidies that are huge and invested in cheap fertilizers, do we really need that? Cheap fertilizers make farmers just put all amounts of fertilizing into the crops because it's cheap. So a lot of it is not needed. A lot of it runs all the way to the coast, and you get a coastal dead zone. So instead of putting that money in something which is really bad for the environment and really bad for the farmers too, it's only good for the companies, that money could be used for positive incentives for agroecology, so to speak. I'm just giving an example. There are many ways in which we can keep having a good life or even a better life without damaging biodiversity. And there are a number of ways in which we could make it really expensive for all these activities that damage biodiversity, because if you damage the fabric of life, you damage the common good. So that would be my general recommendation.
Thank you so much for all of your presentations.
Yeah, a good ending.
Okay.
I would like to thank all of you for terrific questions and the three speakers for a very good morning.
For your time.